Hello everyone, good afternoon and a warm welcome to this webinar. So we are MedGenome, the leaders in genetic testing in India. My name is Joseph. I'll be your host for the webinar today. So today's webinar is in association with Federation of Gynecologists and Obstetrician Society of India, FOGSI. And this is a three-part series on prenatal testing in association with the Genetics and Fetal Medicine Committee of FOGSI. And I'd like to thank Dr. Mandakini for providing us this opportunity to present to this audience. And so the topic for today's webinar is chromosomal microarray and its utility in prenatal screening. And the moderator for today's session is Dr. Priya Kadam. Dr. Priya is the Associate Director for Reproductive Genomics at MedGenome. She has completed her MBBS from Bangalore Medical College, and she has a Master's in Science by Research from National University, Singapore. She has also filed a number of patents in the US and China for her work in prenatal testing. She has published in journals such as Journal of Proteomics, Cancer Biology, and many others. So she's been associated with MedGenome for the past 6 years, and she is in charge of prenatal testing at MedGenome. Before I hand over to Dr. Priya, I would like to inform the audience that if you have any questions or comments, please use the Q&A window of the Zoom conferencing interface. We will address the questions at the end of the session. And we will also be sending out a recording of this webinar to all those who've signed up. So look out for it in a week or so. And so now, without any further delay, I will hand over to Dr. Priya, who will take this session forward. Over to you, Dr. Priya.
Thank you, Joseph, for the kind introduction, and a warm welcome to all the panelists and speakers as well as the participants. It's a pleasure having all of you on board, and it's my responsibility to introduce the most distinguished chairpersons, Professor Narendra Malhotra and Dr. Archana Basser. I think Professor Narendra Malhotra needs no introduction. He has several achievements to his credit. He's been the past president of FOGSI, and also is the managing director of Global Rainbow Healthcare and Art Rainbow IVF. He has held very senior positions and top positions in several societies. And he's also the recipient of several awards, including the Nepal Samman Man of the Year Award, Best Citizen of India Award. His several publications as listed here, and also as an editor of several books. And with that, I'd also like to introduce our second chairperson, that is Dr. Archana Basser. She is the Vice President of FOGSI 2020 and also the Organizing Secretary of AICOG 2021. And she holds a Guinness World Record for removal of the largest uterine tumor. So over to you, chairpersons, to say a few words before we begin the sessions.
Very good morning, good afternoon to all of you who are attending this. It's indeed so heartening to see that genetics has been so nicely being discussed in our webinars, and especially testing, because a normal gynecologist, when he just writes karyotype. And when the report comes, and probably some of them don't write FISH or karyotype or microarray and don't understand what they. So it's just writing a test and knowing what you're writing for, and knowing what information it's going to give you, and then planning the future pregnancy or infertility or even cancer studies is very important. So genetics does not involve anomalous fetus or looking for chromosomal defects or looking for it. It involves a whole lot of stuff like male infertility, female infertility, genetic basis of cancers, genetic basis of disease, whole lot of non-communicable disease which might have a genetic basis. And we are born with all sorts of genes. We are born with chromosomes. On the chromosomes, there are genes, there are gene loci, and there are thousands and thousands of genes. And these genes are responsible for what we are. And now they have been decoded with the whole genome project. And with a simple test, you can probably find out in yourself what are the good genes and the bad genes. But it's not only that diseases has a genetic basis, it's also the epigenetics. So you may be having bad genes or cancer genes, but they might lie dormant if your lifestyle is absolutely okay and good, and you're not triggering off the bad genes and the good genes. So it is a matter of genetics, which is the hardware, and epigenetics, which is the software which triggers off these genes. So indeed, congratulations, Dr. Mandakini Prahar, fetal medicine chairperson of FOGSI, for hosting these great series. And we look forward to each and every one of these series. And thank you, MedGenome, for bringing this to us on a good platform. It's indeed a pleasure for me to be here as chair of this meeting.
Hi, good afternoon, everyone. At the outset, I bring greetings from FOGSI and Fetal Medicine and Genetic Committee of FOGSI, which is under me. I am proud to be here on this webinar, which is being organized by Dr. Mandakini Pradhan along with MedGenome. I think most of the thing Dr. Naren Malhotra has already said. I really endorse that genetics is very, very important. As a common gynecologist, when some problem comes to us, we think, oh, it's a genetic problem. We don't know. But it is important that you have to understand what the genetic problem involves, and you should know where properly to refer them and what investigations are to be advised, so that we can in turn help the couple and the progeny as well. So it is a very important series of webinars which Dr. Mandakini has taken in her hands to educate and to sensitize common gynecologists who are not practicing fetal medicine to understand what all it involves. It's not only, as said already, chromosomal disorder, it already that it is the genes are the basic foundation of the being human being. So we have to understand it in detail. So congratulations, Dr. Mandakini Pradhan and MedGenome for providing the platform. And let's begin with the scientific program. And thank you, Dr. Priya, for the kind reading of the scene.
Thank you, chairpersons. Over to the next slide, Joseph. Yeah, Professor Mandakini Pradhan will be our first speaker. She will introduce genetic tests and also what is genetics and genetic tests. And Professor Mandakini Pradhan again needs no introduction. She is the professor, and but I'll just mention she is the professor and head of Department of Maternal and Reproductive Health, SGPGI, Lucknow. She is the chairperson of Genetics and Fetal Medicine Committee, FOGSI. She has several achievements in her kitty. She has publications in several index journals. She's been quoted in the William textbooks. She's been awarded Bangladesh Ultrasound Society Award in 2019, as well as Nari Shakti and Bharat Gaurav Samman in 2020, and many more. So over to you, Dr. Pradhan, for the session.
Good afternoon, everybody. I thank FOGSI for giving this opportunity to make me becoming chairperson, and then simultaneously getting this platform to get connected to people who are all over India, and to make people aware about what is genetics. And I'm thankful to Dr. Archana Basir, always encouraging, very, very encouraging. The moment I write her, if I can do this webinar, within 1 hour, I get a response that she's always in her toes, that yes, you can go ahead. So never ever I have heard a no from her. And Dr. Dr. Narendra Malhotra, sorry for that, interchangeable. But then you are such a person that has encouraged people to know what is the upcoming things. See, 10 years back when I was about to join genetics, people started saying that people are dying of diarrhea, you are going to do genetics, what will it will happen? That was in 1997. But then now, probably without genetics, our day-to-day practice will be difficult. So to educate everybody, to learn something from others, we have planned this series of webinars. And the first one will highlight chromosomal microarray. But then before going on to directly jumping onto microarray, I'll be taking you through why should we know at all genetics? We should not be this 20th century. Now we should be looking like this and thinking about, we probably should know about genetics. The whole program is sponsored by MedGenome, that I declare. So genetics has come all the way from Mendel's era of P experiment, that is tall and then short P and then 64 characters of P that he discovered, and he probably identified the inheritance in human. to Human Genome Project that has been encoded. There is no conflict of interest that I need to declare. So the progress of science has led to from Mendel to a Human Genome Project and people thinking about a designer baby. Modifying this gene and that gene and making a baby, say, without abdomen, what is depicted, probably not possible, but then still people thought process cannot be stopped. There is progress in the society and thereby there is less financial issues that we used to come across as a student or in earlier days. If the problem can be solved with money, then it's not a problem. People can arrange money. And there is increased awareness and thanks to our internet. So the pregnant woman may accompany not by her husband, but a lawyer to you. So there is increased awareness. People start investing for their health care. So in genetics, in routine practice, where all we really fumble is, if we detect a malformation on ultrasound, malformation in the delivery room. Previous child with malformation, the couple has come to you for subsequent pregnancy management, or a baby with mental retardation, recurrent pregnancy loss, inherited cancers, infertile couple. The other day I was, Dr. Niharika was conducting a webinar on infertility, genetics of infertility. It was really, really interesting. And that made me put one slide at least of infertility. Then primary amenorrhea, screening for maternal and fetal diseases. All of us are jumping onto double marker, triple test, cell-free fetal DNA. Without knowing it, without understanding it, it will definitely be difficult to manage the patient or the couple in a proper way. So these are all few malformations that can be detected in antenatal period, first trimester till third trimester. And in our country, we still see neural tube defect being diagnosed in the third trimester of pregnancy. And here it is a double bubble and a hollow prosencephaly with a proboscis that can be detected, or a hydrops can be detected in the first trimester. So malformation at birth, this fetus, the photograph of the fetus was taken by the couple and the baby was alive for 2 days. So, and then this was the hollow prosencephaly fetus, and this is anencephaly along with midline fusion defect. All the organs are outside the abdominal wall. So malformation may be detected at birth, or the couple may come to you with a previous child. Say, for example, this child, if you see this, the video of this, see, there is difficulty in getting up of this child, and you definitely, if the child is asked to get up, he's climbing on his knees, and that is muscle dystrophy. Or the female may come to you with a child with Down syndrome and she being pregnant, and she wants to have a healthy child, not like that. The patient brother is affected with some mental retardation, whereby absolutely severe mental retardation. The mother is accompanying the sister. The sister and her brother have come to us with this. If you leave the hand of the child, he starts biting. See, and it is obvious that this child is mentally subnormal. And then this is previous 2 children affected with, say, hearing loss. So if somebody is asking her name, but then the mother once ask her about in the sign language, then she starts writing it. So they are not mentally subnormal, but they are, both of them were deaf. So previous 2 children affected with some abnormality. These are common clinical situations we come across day in and day out in our routine practice. So infertility, should we go and start doing genetic test, or is there a genetic origin of inheritance of infertility in a couple? So in male infertility, genetic factor contributes commonly, whereas female infertility, it is less common. And in recurrent abortion, 5 to 6% of the couple may be a carrier of chromosomal abnormality. But about azoospermia or oligospermia or a normal sperm count, all of them can have a chromosomal abnormality and hence warrants for a chromosomal testing. There is a subset of patients with oligospermia should have a Y microdeletion study, and during this series of seminar you will be cleared about what is Y microdeletion. And there is something called obstructive azoospermia, whereas there is some obstruction and it is because of congenital bilateral absence of Vas difference, and most of them they carry a mutation in the cystic fibrosis gene. So with this knowledge of azoospermia, the patient has to be tested for CFTR mutation, chromosomal abnormality and Y microdeletion. You will be wiser in managing such a patient. Coming on to inherited cancer, I'm just touching upon. All of us know about Angelina Jolie's getting mastectomy done. Her mother died of breast cancer and her grandmother died of ovarian cancer. So is it not inherited? Then by this time, all of us know that it is breast ovarian cancer can be inherited. But what we do not know is, if we see such a patient of ovarian cancer, we do not go ahead and look for any family history of cancer and thereby not testing about it. And the time has come to probably offer that test and interpret that test. How do you manage her daughter or her siblings or any family member with ovarian breast cancer? So basic genetics, once you unfold one of these things, it is never the same. So you start unfolding and it goes on and on. So I'll be taking you through some genes, DNA and chromosomes. DNA is nothing but stands for deoxyribonucleic acid. They are present in the nucleus, in the chromosomes. We have got 46 chromosomes, 23 pairs in each cell, 22 autosomes and 2 sex chromosomes. And what are they? They are the building blocks, or the building blocks of DNA are nucleotides. What we hear about is A, T, G, C are the bases. These bases are combined with a sugar moiety and a phosphate bond and forms this double helical structure that is seen over here. And this double helical structure, what is a DNA? And if I stretch the DNA, or I take out the DNA of a human being and stretch it, it will be going and coming 6,000 times to the moon. So this is so compact that it is compacted into one cell. So this is so hence we need to, why we need to know is, if we need to, we know about these base pairs, so we can understand that a change in base pair can lead to a disease condition. So there are 30,000 genes in human being. Many genes are there in each chromosome. So here is one chromosome. Say for example, I have taken this and have expanded. There is one gene over here, second gene over there. So this small area has got more than 2-3 genes. So a part of DNA that codes for a gene, and a gene encodes a protein. So, but one should know all DNA present in our genome doesn't code for protein. So there are in between areas which doesn't code for any protein. So genome organization can be better understood with a book analogy. And what I have done is, see, if you write words make sentence, and then subsequently you have a book, and then the book is put onto a rack, and the rack is nothing but one chromosome or a rack of book. So alphabet makes words, words make lines, lines make page, and pages make book, and then book are stuck together to make a book rack. Similarly, our ATGC, or what I told you about nucleotides or base, 3 of them forms one codon that codes for one amino acid. Multiple amino acid put together forms a protein, or it is a gene which codes for a protein and multiple genes forms a chromosome. So this analogy I'll be using to explain various tests. Say for example, I can look into this book rack and see if the rack is properly arranged or not. And looking into the book rack and making a report that whether all the books are there in that rack or not, or there is some addition, there is some deletion, then that is known as a chromosomal study. If I am looking into the pages of each book and then trying to see that if all the pages are there in each book or not, then that is microarray testing. If I am going and reading each line of the book and trying to figure out if each line is properly written or not, then that is known as a sequencing. And what is exome sequencing? I'll be coming on to it. So in the reverse order, if I start reading every word of this book, then that is called as a sequencing. Say for example, here is the book which says that every village should have a toilet. And let me take this word toilet. Instead of toilet, I is missing and hereby it is toilet. So every village should have a toilet. So it doesn't make any sense. If it is replaced by omelette, so every village should have a omelette. That means that implies that every village or every person living in that village will eat in one omelette every day in the morning. So the from toilet we have come on to omelette by changing TOI to OM. So by changing 3 words, 3 letters, the whole meaning of the sentence changes. Similarly, by changing 1 or 2 base pair of a sequence of the gene, the whole meaning of the protein or the protein coded for that gene changes. So hence, and causing the abnormality in the baby. So genetic test is chromosome, then microarray, and then what I said is sequencing. But what is microarray is, for example, you look into each page of the book. Say for example, you found out that 49 page and after that it is 50 page. Or you found out that 50 followed by 50. So there is a deletion of 50 or duplication of page 50. So a deletion or duplication of a page of a book is detected by microarray, or deletion or duplication of a part of the chromosome detected by microarray. So microarray testing tells you about deletion, duplication in the genome. So chromosomal abnormality, maybe these are normal chromosome, that is I have depicted with a book rack, and there is some deletion in this chromosome which is so obvious and we can detect it. So abnormal, that is detected by a karyotype or chromosomal analysis or looking at the book rack. But if there are some book page is missing, can I detect with looking at the rack? No, I cannot detect that looking at the rack. And abnormality not detected by karyotype or the missing of the page of a book is tested by doing a microarray analysis. So microarray analysis is nothing but looking into the pages of the book. So chromosomal abnormality or the rack number of the racks that are present in a library or in your genome, in other words, can be, say 46 racks are there in this library or 46 chromosomes are there. If you add 1 rack or the rack number 21 is added, so in other words, there is addition of a total set of books of that rack or a whole set of chromosome, that is known as trisomy, or it is called as, because it is 21 chromosome is added, it is called as a trisomy 21 T21 Down syndrome, that is we commonly call it as. So chromosomal arrangements are in the genome, they are not arranged the way it is seen in the picture. It is arranged. The books in the library are arranged by the librarian. Similarly, the books in this are arranged by the microscope or by testing. And this testing is called as a karyotype. So the books are lying like this, or the racks are lying like this, and it is tested, or in other words, it is in a cell, the chromosomes are lying like this, under the microscope, and under the microscope it looks like this, but it is arranged by the test, and after arranging it looks like this. So arranging of this and figuring out whether they are normal or abnormal is the test, what is called as a karyotype. So karyotype is looking onto your the book rack, and it is done by taking the dividing cells. So blood is collected in a heparinized vial, the lymphocytes are isolated and they are cultured. So they start dividing. They are arrested in metaphase by adding some chemical and spread onto the slide. Staining is done and observed under microscope. So why do you should not go ahead and do these tests, but why do I have this slide is, you should know that karyotype can be done on live cells. So hence, for sending a sample to the lab, you need to collect blood in heparinized vial and the cells should be live. Because if you start sending product of conception and the product is a product of missed abortion, all the cells are dead. So you will not be getting any live cells and hence you will not be getting a report for this. Next is this numerical abnormality may be detected in spontaneous abortion. Never think that it is only by doing a double marker or triple test that or quadruple test it will be done. It can be detected by screening antenatally. A child may be brought to you with Down syndrome, and as reported by me, fertility in a man with Down syndrome, a patient may attend to your infertility clinic and on examination may be found to have Down syndrome. Similarly, trisomy 13, an excess of chromosome 13, or there may be a deletion of one chromosome or absent of one chromosome, whole set of sex chromosome is absent, then hence it is called 45 because one chromosome is less, and XO means one X is there, the other sex chromosome is absent, and we commonly known as Turner syndrome. All of us have seen primary amenorrhea, short stature, and variable other clinical features. A whole set of chromosomes may be in excess, that is 69 chromosomes, and is called partial mole. The placenta will be very thick, and baby may be growth retarded. Early onset growth retardation without any obvious malformation may be seen in such a pregnancy. So these are all chromosomal number abnormality, or you have a either excess number, that is trisomy or triploidy, or there is a less chromosome that is known as monosomy X, or you may have a structural abnormality in the chromosome, or the chromosomal structure is abnormal. There is a translocation between these two chromosomes. Part of it has gone, pink has gone to purple and purple has come to pink, or one chromosome has gone to other, that you commonly see in a couple where we look for recurrent spontaneous abortion. And it is balanced, and hence it is called as a balanced chromosomal abnormality. And if there is deletion or duplication of it, then it is called unbalanced, or there is some loss or gain of genetic material, and hence it is called unbalanced. And unbalanced usually will have some amount of clinical features. So now in this, again coming on to this library or chromosomal library, if just see these two chromosomes, part of this book has gone on to anatomy book, has gone on to pathology rack, pathology book has come on to anatomy rack, but there is no loss of any book or any anatomy book or gain of any pathology book. So there is no loss or gain of material of or the books, and hence it is called balanced reciprocal translocation between two chromosomes. So translocation between chromosome 2 and 10, and this is how it is written. So in this pedigree, this couple has come to us with previous 5 abortion. And what is the finding? The finding is there is a balanced translocation between 4 and 12. And this translocation between 4 and 12 is the cause of recurrent spontaneous abortion. So what will happen to their gametes? The gametes will be either the gamete will have both 4 and 12 normal, or 4 and 12, which is translocated. Thereby, there is no loss or gain of material or chromosomal material, and hence it is balanced carrier. But if rest of the chromosomes are normal, but if the other way around, if the or anatomy book is there, but there is loss of pathology book over here, and there is gain of anatomy book over here, so then there is loss or gain of material in the gametes, and hence resulting unbalanced gametes and resulting in abnormality. So the translocation can be reciprocal or Robertsonian. The whole chromosome can go and got attached to an acrocentric chromosome. Here in 13-14, there are 213, and instead of 214, 114 has gone and got attached to 13. So it is called as a Robertsonian translocation. It can be between two same chromosome, that is 21-21. The same number can, one goes and gets attached with the other. So that is, and why should I know about it? If it is different chromosomes, say 14-21 translocation, then the gametes will be, it can have a balanced carrier chromosome, can result in normal, or can have a normal outcome in pregnancy. But if it involves the same chromosome, that means there is 21 has got on and got attached with the 21, then what will happen is if it is different, then it can have normal or balanced carrier, or can have an unbalanced gamete. But if it is the same chromosome, the gametes will be always unbalanced. So both the 14 will go to two gametes, but this attached 21-20 either go to one gamete, and the other gamete will have nullisomy for chromosome 21 and disomy for chromosome 21. So hence all unbalanced gamete, and in such a patient there will be recurrence risk of 100%. Here, as a IVF specialist, Dr. Mehrotra will use the donor gametes. If it is the female who is carrying it, and hence the donor ova should be used, otherwise they will not have any success pregnancy outcome. So hence what we need to know, friends, is if it involves two same chromosome, then there will be always the fetus will be abnormal and will require a donor gamete. Whereas if it involves two different chromosome, then almost 90%, more than 90% chance that they will have a normal pregnancy outcome. So this is what is karyotype and what are the indication, or looking into chromosomes is what our indication is recurrent abortion, high risk for trisomy 21 on screening. If there is dysmorphism or malformation, all patients with mental retardation is the first workup. You go ahead and do a karyotype. If a baby is born with ambiguous genitalia, first thing you look for what is XX or XY, and then rest of the karyotype. Primary amenorrhea, always we go for. Premature ovarian failure, probably in the course of our course, we need to highlight on this. Male infertility, neonatal death or stillbirth. These are indication of doing karyotype. And microarray analysis, as I explained you, and today's seminar we will be highlighting what are indication. But one must remember that if there is a balanced chromosomal rearrangement, microarray cannot detect it, and it cannot detect single gene mutation. But deletion duplication, yes, it can detect it. What are the indication? Fetal malformation, if your karyotype is normal. Stillbirth, you can't do a karyotype, then you go ahead and do a microarray. You are doing an invasive testing for something else, so you do a microarray so that you offer to have a microarray. Mental retardation with normal karyotype, however, you need to have pre-test and post-test counseling. So coming on to reading the book, the sentence of the book. In this library, can I read all the books present in the library? No, it's not possible. So I need to have some direction which book to open. I'm interested for obstetrics. I will go ahead and start reading obstetrics and find out if there is a abnormality. Similarly, if I give some clinical information and the computer tells me what all genes that is responsible for that clinical manifestation, then those genes are looked for from A to Z. is called clinical exome sequencing. For example, hearing abnormality, there are say 50 genes, 52 genes, they look for that. If you inform the lab that the child has got developmental delay, polydactyly, etc., etc., they will put into computer, they get a set of genes, those genes are looked for. Similarly, in skeletal dysplasias, a set of genes are sequenced, that reduces your cost and timing of this thing. So we normally these days we are getting confused with whole genome sequencing, whole exome sequencing, and what is clinical exome sequencing. Our subsequent lectures will be on this. The last series is on exome sequencing, but reading only the normal labor from Williams textbook, exactly from Williams textbook, is looking for only mutation analysis for a single gene disorder. If I go and start reading the whole of obstetrics, then that is clinical exome sequencing. If I start reading the meaningful books or on the informative books, say Williams plus D Suite and more, then that becomes clinical exome sequencing. If I start reading the all books present in the library, then that is whole genome sequencing. So you can imagine whole genome sequencing is clinically may not be relevant in this era. So indication of exome sequencing is helpful if karyotype microarray are normal and the child is abnormal. Test the affected child first. If not possible, the previous child is not living, then you get some information and test the couple. One couple can be tested if specifically if the condition is autosomal recessive in nature, and then the same can be looked in the other couple. Other investigation may guide you, say previous child, any other investigation like MRI or any photograph or anything, better to see the couple in preconception period. Coming on to single gene disorder, it is like knowing about cardiac output in pregnancy. I will go to obstetrics rack, take out Williams textbook, read the chapter 4, 49 to 79, page number 61, gives me the answer about cardiac output in pregnancy. So I read that. Similar to this analogy, the single gene disorder is clinically disease is identified, the defect protein and its gene is known, same gene is tested. For example, beta thalassemia. Clinically, we know that the child has got beta thalassemia or the couple is carrier of beta thalassemia. The defect is in beta globin gene. It is present on short arm of chromosome 11, and look for mutation in that beta globin gene only, or reading only page number 61 of the Williams textbook. Or you can sequence the whole of beta globin gene, that is 49 to 79 page number. So these are two methods to look into single gene disorder. We'll be coming on to it. This HPLC indicates that the one of the couple is beta thalassemia carrier. So clinical examination like neurofibromatosis or Meckel-Gruber syndrome, by examination of the previous child, we order the lab to look for the specific gene, that is neurofibromatosis, or gene for Meckel-Gruber syndrome. Or you may suspect the gene and the defect, that is previous respiratory distress, children dying of respiratory distress. Think about spinal muscular atrophy or cystic fibrosis. The couple can be tested easily for it. Previous ambiguous genitalia, think about congenital adrenal hyperplasia. Previous short limbs, it could be any of the skeletal dysplasias. Previous baby requiring blood transfusion, dying around say after 6 months of life, say 1 year, 1.5 years. Think about thalassemia and test the couple for it. So the genetic test is karyotype or chromosomal analysis. They are done on live cell. If you are sending a blood sample, you need to collect it in heparinized vial. And it is usually, we order it in recurrent spontaneous abortion or in fetal sample. Then fish, I have not touched upon, we will probably touch upon. It is for specific chromosome, that is chromosome 21, 18, 13, or specific microdeletion that we suspect by clinical examination. Microarray can be done on any tissue. When chromosomes are normal in a malformed fetus, that is the indication of doing and going ahead and doing a microarray. They will tell you if the page of the book is missing or not. Clinical exome sequencing, say on the affected child, affected mental retardation, skeletal dysplasias, say skin abnormalities, specific known disease, then mutation for that disease to be done. So that's an introduction to what all genetic tests we come across and we'll be coming across in coming days and are readily available in India. Please do not have this conception that this is not available in India. And these are every day, the cost of these tests are decreasing rapidly. So we need to know about it, ask the patient to do it and interpret the result. The lab these days, they usually help us in finding out or interpreting these results and guiding us what to be done. So I thank all my patients for making me richer by their suffering. All faculties and residents of my department, the molecular laboratory that always supports, and FOGSI for making me coming to you and making my thought process going to you. So I thank you all for having your patient hearing. We look forward to all these series of talks today and in subsequent lectures. If you have any difficulties anytime, you are always asked to put it in question answer session. You will be definitely answered. That will be more helpful rather than having only one-sided talk. Thank you all, and thank you MedGenome for allowing us to hold this. So I end my screen share so that everybody can see it.
Thank you, Dr. Mandakini Prahar, for such a fantastic talk, especially making it so simple for us to understand how chromosomes and genes are organized by just giving the example of books in the library. It simplifies so much and makes people understand how we can understand what is a gene, what is a base code, what is a codon, and what these tests which we have, like FISH, karyotype, microarray, whole genome sequencing, exome sequencing, next generation sequencing, when they should be ordered for what indication, and what do they tell us. So any questions? I don't see any questions yet in the chat box. I think there is one now. No, there will we be discussing about recombinant DNA technology also? No, I don't think this is a recombinant technology workshop. It is just about genetic testings. Two participants are raising their hand, but they have not put their questions. Please put your questions, because otherwise, if I
So actually, I think there is one other question. So maybe I can just ask you right now, if we see
can you stop sharing your screen? It's still coming.
I'm struggling to do that. yeah, now I got. Resume new share. So it says new share. It doesn't say resume share. What do I do?
I've shared.
I think,
okay.
Yeah.
So, yes.
So I think we'll just ask you one question that is here. If we see an abnormality on karyotype, do we stop at that step? That is the question.
Yes. Yeah. Once we see an abnormality in karyotype, we should stop at that stage and the defect is identified. There is little catch of it. If, say, whether it is balanced or unbalanced, that can be picked up by doing a microarray. So you have a clinical examination. If the couple is absolutely fine and they are carrying a translocation, then you think that they are most likely it is a balanced translocation and you need not do any further testing. If they have some manifestation, then you need to find out which are the genes which is duplicated or deleted in that translocated site. Then you need to go ahead and do a microarray analysis. That will pick up your deletion or duplication.
What test will you do for thalassemia triad and major? That is another question. and comment on NIPS, comment on the cost of these tests.
Cost of this test varies, but definitely these are doable. Say, for example, karyotype cost around say 2,000, 2,500 rupees.
So they're not very expensive these days. They're
not very expensive these days. The highest expensive test is probably the clinical exome sequencing is say around 25,000 or so.
25. So that's the maximum.
So then that is, and single mutation depends on the disease.
So what test will you do for thalassemia triad or thalassemia major?
For thalassemia major, you need to ask the lab to do a mutation analysis for thalassemia major. It is as simple as this. Please, friends, for any other test, you please write mutation analysis for, say, for example, if you tell your clinical manifestation to the lab, you will get a phone call from the lab saying that you have written the baby needs, say, blood transfusion at 8 months of life, 10 months of life, looks pale, hemoglobin 2, whatever you write these. So they will definitely interpret and do beta globin gene sequencing for you. So it is mutation analysis for beta thalassemia.
So thank you, Dr. Mandaki, for her. I think we can go to the other one, Dr. Kadam.
Yes. So there are a couple of other questions which we will bring up later. Yeah, we will bring it up later.
We'll take it in the end.
Yeah. So we have, now we have Dr. Shaktivel Murugan. He's the Associate Director of Lab and Operations MedGenome Labs. He has a PhD of Human Genetics from BITS, Pilani. He has several peer-reviewed publications in human genetics, and he has more than 10 years experience in advanced genomics and molecular biology techniques such as NGS and microarray. He has been trained with Illumina and Affymetrix. He has sound knowledge of application of this technology, and he manages the lab operations of the largest multi-department genomics company in India. So over to you, Dr. Shakti.
Thanks, Priya. Good afternoon. So my presentation, it's going to be on chromosomal microarray. So I'll be talking on the basics of microarray, a little bit on the technology, and the interpretation of results, a little bit on that, and its diagnostic utility, both in terms of prenatal and postnatal testing. So just this is just an outline of what I'm going to talk now. It's briefly about MedGenome, just a couple of slides, and a little background on chromosomal microarray and copy number variations, and comparison of different technologies, indications for testing, advantages and limitations for chromosomal microarray, MedGenome data, some case studies, and conclusion. Yeah. So I'm sure, I'll just rush through these slides. We are the largest genomics-based diagnostics and services company with the largest output in terms of capabilities, throughput capabilities for next generation sequencing. We cover also other genomic tests like prenatal testing like NAPT, PGS, flow cytometry, immunohistochemistry, so all bases covered. We have end-to-end solutions, as I told you, starting from Sanger, IHC, microarray, MLPA. So, and coming to next generation sequencing, we have the largest throughput lab in India. We are also spread across in Singapore and US, where we don't do diagnosis, more of research services. Our operations go this way. There's sample accession. It can be different types of sample depending on the test. Samples get shipped to lab where we do processing, where nucleic acid extraction, library preparation, or any enrichment test steps happen. Then sequencing or any other test or real-time PCR-based test, data generation happens. Analysis of raw data, processing analysis, and genotypes and phenotype correlation and reporting happens. Interpretation of the data leading to a report. A report is shared with the clinician. So this is the regular, operation workflow for most of the tests which happen, genomic tests which happen here. And coming directly to the background of what chromosomal microarrays and copy number variations are. Just to give a background of genetic disease burdens, this is a publication by Dr. Dr. I.C. Verma. Very high proportions are congenital malformations and chromosomal diseases. High numbers of trisomies, the aneuploidies, like Down syndrome trisomy and 13 and trisomy 15. And there are high numbers of beta thalassemia, ACD. So all this, adds to the burden of diseases among the prenatal conditions, and testing becomes very critical. Prenatal testing becomes very critical to understand whether the children are going to be born with some disorder or not. And there are a lot of new technologies for the same, and but with so many technologies, we are not sure which one to use for what condition. So we, as Madam Dr. Mandakini put it in her last presentation, there are several tests available now, starting from karyotyping to FISH to microarray to next generation sequencing, targeted sequencing. What are we going to use when we see some malformations in the ultrasound which we take during prenatal testing? So there are other challenges for this, like their clinical phenotyping is not really clear here. We need to be really careful about what to look for, even in the test we are doing. And it cannot be assessed, many of these traits cannot be assessed in utero. And comes the challenge of maternal cell contamination, especially with prenatal cases where we collect amniotic fluid and CVS, which are the commonest samples. There is a high chance of maternal cell contaminations, which can affect the outcome of the results. And add to this, very rapid turnaround times are crucial, very crucial, because most of these samples comes for testing only around after 16th week. If we have large turnaround times, it is very difficult to take a decision on the baby before the 20-week deadline, which is being given by the government. The issue, the mutations or the changes in the genome can be at different levels, and based on what level or what size of the gene is affected or the genome is affected, different types of tests can be used. The scale, it can be at a chromosomal level, where large whole chromosomes are duplicated or deleted, which are called aneuploidies. Sometimes there is a large part of the chunk of the chromosome which is affected, which can be picked up by karyotyping or FISH. And sometimes it is only small size regions of the genomes which are affected, anything from a few kbs to a few mbs, which is very difficult to pick up by using karyotyping, because the resolution of karyotyping is slightly low. And comes the single gene issues, single base changes, which can be checked only by Sanger sequencing. When we look at large changes in the chromosomes, like aneuploidies or large copy number variations, deletions, duplications, there are 3 common tests which we use for testing, and they are, other than the cytogenetics, they are FISH, array CGH, and microarray based, SNP based microarray. So FISH, array CGH, and microarray they are molecular cytogenetic testing, and cytogenetics is more of at a broader level. What is copy number variations? They are basically structural variants involving alterations in the number of copies of regions of DNA. So by definition, anything more than 1 kb in size is considered to be a copy number variations. But the challenge is when a large amount of copy number variations which are present in the humans are polymorphisms. And if you look at the general population or the control population, 5 to 25% there is a regions of genome or copy number variable because of their complexity, and many of these are normal ones. They don't cause a disease. even large copy number variations are known to not cause this disease. So however, the challenge comes when you identify a copy number variation and whether how are we going to interpret it. And this is where the main challenge is, because just like in SNVs, we don't have databases for copy number variations as of now, and the databases are slightly getting better, but it is still not as comprehensive as we have for the SNVs. So there are a lot of interpretation challenges when it comes to copy number variations. Just some a pictorial representation of what a copy number variation could be. So compared to the normal compared to the normal two chromosomes in the center here, if you look at the left, there is a loss of the region C region, which which is called a deletion, or to the right there is a gain of D region, which is called a duplication. And the sizes, as I told you, can vary from a few kbs to many MBs also. So many of them are not pathogenic. So especially when you consider, though by definition we say more than 1 kb is considered as a copy number variation, from the reporting point of view, we don't report copy number variations which are lesser than 100 kbs in size, unless they are overlapping with from some known OMIM genes. So they can also be called as microdeletions and duplications, and usually they are undetected by karyotyping. So the usefulness of microarray comes when you don't identify any lesions and by doing karyotyping, and you suspect a copy number variation to have caused the disease. What are microarrays? So microarrays are just, they are simple silicon slides or glass slides upon which thousands of probes are being, hybridized. So there are a lot of probes which are fixed to those glass slides, and these probes are specific for regions within the genome. And when you actually fragment the genome and do some modifications to it and allow them to hybridize to these probes, only those with the complementary sequences bind, and the rest gets washed off. And we look at the bound region. So these are for mostly, say the probe is targeting a SNV. When only if the SNV is present, the DNA will bind to the probe, and once you wash it, the rest of the unbound DNA gets washed off, and you are able to identify whether an SNV is present or not. Okay, so this basically is based on fluorescence based assay. The probes are fluorescently tagged, and only if the binding happens, they fluoresce. And depending on what base it is, the color of the fluorescence or intensity of fluorescence also varies. And because these are on slides, and because these are you can tag a lot of probes on a particular small region, this becomes a high throughput assay as compared to fish or the MLP kind of assays, where only 1 or 2 lesions can be targeted. So this is a picture of a gene chip, the Affymetrix gene chip. And if you look at that center region, that is the glass slide where the probes are being bound. And Affymetrix gene chip, you have anything between a few 100,000 sequences to millions of probes, to millions of probes which are bound to this glass slide, and you can identify so many SNVs in that. So a gene chip or Affymetrix gene chip is basically a SNV microarray. There are other type of microarray, like from Agilent you have the CGH or comparative genomic hybridization microarray. So the principle slightly varies between Affymetrix SNP gene chip and CGH gene chip, which I'll be telling you in the upcoming slides. There are different types of microarrays. You can actually target anything you want to study. It can be a RNA, it can be RNA probes, it can be probes for proteins, it can be resequencing arrays where you know the sequence and you target only those sequencing bases. There can be a comparative genomic hybridization, SNP arrays, antibody arrays, and tissue arrays. So depending on what you really want to target, you can actually change the type of array. And comparing the microarray technology with FISH and cytogenomics, the cytogenomics, the resolution is minimum of, the classical cytogenomic genetics resolution is minimum of around 5 MB. So it's more subjective, and it's global survey of all the chromosomes, but it's more subjective, and it will be able to detect only large imbalances. Whereas in FISH, this also can detect smaller or larger imbalances. The only challenge here would be that you should know what you're looking for, and you should design probes only for those genetic lesions which you are looking for, and you cannot multiplex too many probes in one assay. So there can be, you can identify an aneuploidy, you can identify translocations, you can identify balanced and imbalanced translocations, but the only challenge here is that you should know what you are looking for, and the sequence should be known. Whereas in the microarray or the molecular cytogenomics, your resolution is very high. You can go up as low as 50 kilobases. So copy number variations as low as 50 kbs can be identified. And another advantage here is that it is not targeted, it is for the whole genome. So mostly the probes are spread out throughout the whole genome, especially with a bit of concentration towards regions which are known to be affected by copy number variations. And I'll be giving the disadvantages of this method. One of the main disadvantages is that it cannot identify balanced translocations as opposed to the other two methods. As I told you, these are all high resolution. This is the typical picture of what you see when you process the samples on Affymetrix assay. You can detect duplications and deletions as low as 50 to 100 kb. And it's used for a wide range of human disorders, including neurodevelopmental disorders and congenital anomalies, such as cardiac defects. I had already mentioned that there are two types of microarrays, the CGH based and the SNPs based. CGH is comparative genomic hybridization, where you mix patients as well as reference samples together and hybridize. If the number of copies between the reference and patient, reference usually has 2 copies. So if the patient also has 2 copies, you see yellow signals. But if there is an increase of copy number of the patient, then you see green signal. And if there's a reduction in copy number of patient, then the reference will take over, you see red signal. So using this principle, you will be able to detect whether the patient has a copy number variation in terms of whether it is a deletion or duplication. And you can, this case, you can do it for the complete genome. Similarly, in the SNP array, it's slightly different. You don't look for copy number variations directly, you look for SNVs. So you type SNVs across the whole genome. And it is well spread out across the whole genome, and the resolution will vary depending on the number of probes you use to type on these microarrays. So if based on this SNP calls, whether it is AA, BB, or AB, so if there is a continuous calls, homozygous calls, it can be it is usually considered as a deletion or homozygous deletion. And if there is a increase in intensity of the calls, it is a duplication. So there's a slight difference between a long contiguous stretch of homozygosities and a deletion, which the software will take care of, and it will be able to call whether it is a deletion or just an LC contiguous stretch of homozygous, LCSH. So the difference in the techniques and what they can achieve is showed in this slide. So as far as aneuploidies are concerned, all these methods are equally good and in picking up. But when it comes to balanced rearrangements, only conventional karyotype and FISH can detect. The MLPA or CGH array or SNP array cannot detect balanced translocations or balanced rearrangements. Whereas unbalanced rearrangements can be detected by conventional karyotype or FISH as well as the microarray. Here, however, you will not know, you will not be able to see it as a translocation. You will still see it as a gain or loss because the translocation is unbalanced. And the triploidy can be picked up by the SNP array, but not by CGH array. And AOH can be picked up by SNP array. All CNVs, whether it is small or large, depending on the number of probes we are targeting, it can be picked by FISH, MLPA, CGH, and SNP arrays. This is just a slide to show what are the different types of array and what they can actually do. This is from Affymetrix. You have 3 different types which are commonly used for clinical, or we use for clinical. You have the Cytoscan Optima, the Cytoscan 750K, and the Cytoscan HD array. So then the difference between each of these is the number of SNPs which are targeted by them, and eventually the resolution of the arrays. So the Cytoscan Optima targets only around 18K SNPs, or sorry, 148K SNPs, and it has got only 18K probes. And because it's only 148K SNPs, the resolution is a few hundred KBs. Whereas Cytoscan 750K, the number of SNPs are around 200K, and your resolution increases. I mean, you can detect copy number variations as low as 100K or even less. And when it comes to HD array, the Cytoscan HD array, since the number of SNPs are targeted or more there, your resolution increases to as low as less than 50 KB in size. This is a software which we usually use along with the Affymetrix arrays. It is called the Chromosome Analysis Suite, where the raw data will be uploaded, and you can see the copy number variations like in red and blue as gains and losses. You will also have the ISCA nomenclature for all these copy number variations we are identifying. However, similar to next generation based sequencing, you will have quite a lot of copy number variations detected for each and every individual, many of them being common variants. So you should be able to interpret the data and be able to identify the causative variant from this, and that is the challenge with the lack of sufficient databases. This is just an example to show how a copy number is seen. So as you can see here, there is this is a gain. You can see that as compared to the allele peaks, as compared to the copy number of 0, which is the normal, you can see a deletion, copy number of -1, or a duplication of copy number of 1. So, and that will be reflected in the allele peaks also. So allele peaks basically gives you normally it is only 3 allele peaks should be present, because it is either AA, AB, or AB, AA, AB, or BB. If the number of allele peaks reduces, so if there is a deletion, there is only 1 copy there, so you will only have AA or AB. So that is seen as just 2 allele peaks. If you have a duplication or a triploidy, you will have 4 allele peaks, because it can be AAA or ABB, like that. And this is an example to show the long contiguous stretch of homozygosity, where you see that normally you see 3 allele peaks, but just with a few gaps in between. This is not a copy number variation. It's more of a homozygosity stretch, which is usually caused in consanguinity. The ISCN nomenclature is the common nomenclature used for depicting copy number variations, and even for the cytogenetic findings. So this is where we basically it denotes the genome build which we are using, the chromosome band, which band is affected, and the coordinates, and what is the copy number. So in most of the microarray reports, you will see this indicating which region, which chromosome is affected, and what is the copy number change. If it is a duplication, you can see it as into 3. If it is normal, it is usually into 2. You will get it as 46 XY or XS. But when it is a duplication, you will see it as into 3 or into 4, depending on how many copies are there. As I told you, the interpretation part is the most critical thing here, and we use the ISCA-based guidelines and the ACMG classification criteria for interpreting the CNVs. The CNVs can be interpreted just like in the SNVs. They can be either pathogenic or likely pathogenic, VOUS, likely benign or benign. We usually don't report likely benign and benign variants because they add to the confusion. So only the first 3 types of variants are reported in the reports. So a pathogenic variant is usually a known microdeletion or microduplication syndrome, or it is there a CNV is present in a place where the relevant gene is there, or we do a parental study and we identify it as a de novo variant, or it is previously reported as pathogenic in databases or publications. So if the CNV which we have identified, any of these meets any of these criteria, then we will classify them as pathogenic. For likely pathogenic, it has to be described previously in a single or few patients with similar phenotype, or in a gene with this SNV region has a function relevant to the region of the patient phenotype, but it is not reported before. And VOUs, they are broad category of CNVs which have not been reported, or if you've reported, there is insufficient evidence. So when there is an insufficient evidence, but the size of the CNV is such that we'll have to report it, then we go ahead and report it as VOUS. We usually don't report likely benign and benign variants. Coming to what are the indications for CMA testing? Sorry, this is again the different classification indications. There are two requirements where my CMA has to be tested. One is the prenatal cases, and the other one is in the postnatal cases. So among the prenatal, it has been as per the American College of Obstetric and Gynaecology, when fetal ultrasound anomalies are detected, or when a patient is undergoing an invasive prenatal diagnostic testing, CMA is recommended. So this could be particularly beneficial to patients with clinical indications of abnormal ultrasound findings, abnormal screening tests, family history of a genetic or a chromosomal abnormality that is detectable by microarray technology, or history of pregnancy loss, typically multiple pregnancy losses. So in a prenatal condition, these are the commonest reasons why we do a chromosomal microarray. However, for postnatal cases, there are ISCA guidelines suggesting microarray testing as the first line of testing for the following disorders: the intellectual disabilities, autism or ADHD, and congenital anomalies. So for these disorders, up to 20% of the cases are shown to have microchromosomal rearrangements, and chromosomal microarray is the first line of testing suggested or recommended by the ISCA guidelines. And if warranted by clinical situation, biochemical testing for metabolic disease has been performed and is negative, then we go for CMA. And sometimes targeted genetic testing comes negative. So there is an X-linked, you suspect X-linked intellectual disability, and Fragile X becomes negative, then we can go for chromosomal microarray testing. This is an algorithm used for a prenatal test, CMA testing. So if there is an indication for prenatal diagnostic testing, a normal fetal anatomy survey, then offer a CMA or a karyotype. If there is an abnormal fetal anatomy survey, ultrasound has to be done, and clinical findings are suspicious. There's a high suspicion of common aneuploidy, then either FISH or karyotype to be done. And if it is abnormal, the report is given. If it is normal, go for CMA. If any other structural abnormalities are there, CMA has to be done. So this is published in American Journal of Obstetric and Gynaecology in 2016.
Dr. Shakti, just a gentle reminder that maybe we should finish in another 2 minutes.
Oh,
yeah.
Okay.
Yeah.
Yeah, so I think just a few case studies I can run through. So coming to the diagnostic yield, if you look, it's been published by ISCA also. For intellectual disability, as compared to karyotyping, which is less than 5%, 15% of the cases show positivity when you test by CMA. And for autosomal spectrum disorders, autism spectrum disorders, up to 10% of the cases show a microchromosomal abnormality. And for multiple congenital anomalies, more than 15% show CMA mutations or CMVs. This is a MedGenome data, just to show that, in prenatal cases, around 9.4% of the cases are showing pathogenic, and around more than 10% are showing pathogenic and likely pathogenic mutations. In POCs, we see around 20% of the cases showing pathogenic mutations, and in postnatal also close to 20%, and which is as per published literature. Which genomic imbalance is clinically significant? Mostly the copy number alterations that overlap with critical regions of known pathogenic regions. And if there is a parental testing and family study is done, and if the parents are not found to be having that mutation, so if it is a de novo mutation, they are significant. And if databases report that as significant, it is better, they are usually pathogenic. This is the ISCA guidelines which I was mentioning. So ISCA is the international standards for cytogenomic array. So they have, as I told you, they have made CMA as the first line of test for unexplained DD or ID, ASD, and multiple congenital anomalies. And if when there's a, it is prescribed or asked to be prescribed even before a G-banded karyotype. Yeah. So this is the basic algorithm for clinical microarray testing. If a copy number change is detected, known pathogenic aberration is there, then reported if it is reported in healthy individuals, it goes as a benign variant. If it is not reported in healthy individuals, but it is inherited from parents, we check whether they are inherited from parents. If it is inherited from parents, then they're likely to be benign. But if it is a de novo, then they are probably pathogenic. If it is a known pathogenic aberration, then it is reported directly as a pathogenic aberration. This just to list out the advantages and the disadvantages. It can be done from any tissue or culture or any. All we need is a good quality DNA. Aneuploidies, microdeletion, duplications can be identified. Better definition and characterization can be done because of their high resolution. It's a genome-wide test, and the data is objective, not subjective, as opposed to the karyotyping. And area of homozygosity can be identified with SNP probes, and it is a good interface it has got good interface with of data with the genome browsers and databases, and we can also detect cryptic abnormalities. Where it has got its own limitations, as I've told you, we cannot identify balanced chromosomal rearrangements. Mechanism of imbalance cannot be identified, and mosaicism very low levels of less than 20% cannot be detected. Tetraploidies or other ploidy levels cannot be detected. Point mutations cannot be detected, or even small aberrations cannot be detected, and all mutations associated with the given syndrome cannot be detected. There can be sometimes one copy number variation and one point mutation. We can only identify a copy number variation. So there are some gaps here in CMA testing because of lack of databases, lack of uniformity of platforms, and the lack of knowledge across the clinical group. So these are the difference between the different platforms, the 750K, Illumina, Agilent. So there are different platforms available. All we need to do is select which one is better for the particular disease based on the resolution we require. So the resolution can be anything between 50K to few MBs. Yeah. As I told you before, MCC is one of the challenges. It has to be done for all prenatal samples. If culture is not being done, if culture samples are used, we need not do MCC. But if you're using directly the amniotic fluid or say a POC or the other samples where MCC is suspected, is better to do MCC. And if maternal cell contamination is there of more than 5%, we should not go ahead with the testing. Only less than 5%, we should be able to do the testing. I'll just show one or two cases. Okay, this is a case of behavioral disorder, craniosynostosis, normal motor developmental milestone, but cognitive and speech impairment. There were two normal siblings, but for this case, when this is a postnatal case, when they sent for chromosomal microarray, we identified a deletion involving chromosome 7 with 7p21, the 7p21.1 region, indicating monosomy for this region. So this is the picture. So from the charts, we can actually see which region is affected, and we can see the lesion also as and we can also see the associated homing genes. So the green regions are the genes. So you can see that there is one gene called twist1 gene, which has been reported for this disorder. And haploinsufficiency has been reported. So a deletion of this region or a heterozygous deletion of this region results in haploinsufficiency and hence the disease. This is a prenatal case where a 2-year-old a child showed anastomosis, a sluggishly reactive pupil, and when they tested this child, we identified a abnormal microarray lesion or abnormal result of 784 kilobase per size. So this region is in the X chromosome, and it was suggestive of a deletion. And there is a gene called NDP gene, which has been previously reported in patients affected with Nori disease. So we identified this in the proband, and when the for prenatal testing, when the sibling sample was sent, we tested for the same region or same lesion, and we found that the fetus did not carry that lesion, so it was a normal child. So it is really helpful not only in prenatal, in a direct prenatal cases, but when there is a family history and we have a lesion which we are going to look for, it makes things easier. So these are other examples for cases where we have identified mostly for intellectual disability, for developmental delay, global development delay, and hypotonia, and another case where small congenital malformations were there and delayed development was there. We were able to identify chromosomal microarray lesions or small lesions from around 50 kb size to up to even 300 kb and MB sizes. So to conclude, it's CMA is a valuable tool in prenatal testing and postnatal testing when when you are considering diseases like autism spectrum disorders, and it should be considered every time invasive testing is done. It should be preferred. Moving forward, should be preferred over karyotype and considered as a first line of test if a structural abnormality is found. And pre and post-test genetic counseling is critical and has to be done so that the understanding of the lesion identified and the effects are properly conveyed to the patients and as well as the clinicians. Thank you.
Thank you, Dr. Shakti, for that very informative talk. We are running slightly over time. So let me quickly move on to our next two speakers, Dr. Mary Poona Chako and Dr. Vandana Kamath. They are from CMC Vellore. Let me go one by one. Yeah, Dr. Vandana Kamath is the Associate Professor at the Department of Cytogenetics, CMC Vellore. She has completed her post-graduation MD Pathology from Manipal Academy, and she has also obtained a postdoctoral fellowship in cytogenetics from CMC Vellore. She has completed her advanced courses in medical education recognized by Medical Council of India. She has several publications in national and international journals. Her main area of interest is genetics and medical education, and other areas of expertise include coagulation and hematopathology. I'll also introduce the next speaker, Dr. Mary Poona Chako, who's the head of Department of Cytogenetics, CMC Vellore. She's completed her graduation from Government Medical College, Kottayam, and has an MD Pathology from Government Medical College, Trivandrum. She has been working in Christian Medical College, Vellore since 2005, and her expertise includes hematopathology, transplant immunology, and immunogenetics. Dr. Mary and Dr. Vandana Kamath are faculty. They are involved in interpretation of chromosomal microarrays as used in both prenatal and postnatal diagnosis. So we will have Dr. Vandana Kamath first.
Thank you, Dr. Priya, for those kind words. So I'll start sharing screen now.
Yeah, please go ahead.
Yeah. So I'll be dealing with the, am I audible?
Yes, please, go ahead.
Thank you. So I'll be dealing with some cases which we came across when we subjected the samples of prenatals for microarray testing. So this is just to re-emphasize on what Dr. Saktivel has already made a mention, just to touch upon it again. So coming to the reporting rules of chromosomal microarray, we look at the number of abnormalities, We look at the type of abnormality, whether it's a copy number variation or uniparental disomy. We look at which chromosome it is involved, the coordinates involved. We also look at the size of the abnormality. Most important, we categorize this abnormality. Then we also look at something called homing genes. These are something which are pathogenic genes. After which we do a correlation, a genotype-phenotype correlation, and if required, we go about doing parental studies. So when it comes to categorization of copy number variation, most important is what I've highlighted in red is pathogenic and likely pathogenic. I will not repeat the words what Dr. Saktivel has already made a mention. This is of prime importance, or this is what is clinically relevant. Coming to something which I have highlighted in green, likely benign and benign, if we get a copy number variation which fall under this category, then we actually look at our internal database as well as published literature. And if this variant matches with what is mentioned as benign in the literature, then as a laboratory policy, we do not include this as a part of our report. Now, what is variant of uncertain significance? This is which is something which is sitting on the fence. What do we do next? So I have certain cases wherein I will highlight the role of variant of uncertain significance and how do we proceed further. Coming to the reporting format, we follow something called an International System for Human Cytogenomic Nomenclature, in short ISCN. So as Dr. Saktivel has already made a mention, so this is the human genome build which we refer to, the chromosome which is involved. This is the arm of the chromosome which is involved. This is the region of the chromosome which is involved. These are the coordinates, and this number represents whether it is 1 copy, 2 copy, or more than 2 means gains. Is it pathogenic? What is the size? And the names of the homing genes. Let us start with case 1. So we had a pregnant lady of gestation age 18 weeks whose biochemical screen was normal. On fetal cardiography, she was detected to have tetralogy of Fallot. So this is how the analyzed data appeared. So the chromosome of interest was chromosome 22. So as Dr. Saktivel has mentioned, something which is marked in red is what represents a deletion, means 1 copy of this particular gene on this chromosome is missing. So how is it represented? It is represented, this straight line indicates there are 2 copies. You can see a dip over here. That means 1 copy is missing. That means it is a deletion. So it was an interstitial deletion. It was located on the long Q arm of chromosome 22. I told you the region that is Q11.2, Q11.2. It's an interstitial deletion. It is about 8.3 megabase in size. This particular region, which was deleted, comprised of 97 homing genes, and the finding which we had actually made tetralogy of Fallot was a manifestation of digeorge, which is nothing but a 22q11.2 deletion. The question arises, do we go ahead with parental studies? Yes, we do go ahead with parental studies because a quarter of patients, this digeorge could be inherited from one of the parent. So how do we proceed with parental studies? Do we do a microarray or we adopt other strategy? So we go about doing a FISH because we have already identified that this was digeorge. We just need to look whether this particular deletion is existent in either the father or the mother. So this is a targeted approach. So we go about doing a FISH testing to test parents for 22q11.2 deletion. Now the question arises, then why did not we do FISH for the fetus? So let us understand this. What are the merits of FISH or MLPA, that is multiplex ligation probe amplification? Of course, both these tests have a decreased turnaround time and it is cost effective. Then why did we proceed with array? Now let us also understand the drawbacks of targeted approach. Let us look at these facts. All cardiac defects are not 22q11 deletion. All digeorge do not present with cardiac defects. In fact, 22q11 deletions account for approximately 5% of all cardiac defects. So some way our questions have been answered. There's another entity which needs to be emphasized on. 10% of digeorge do have atypical lesions. What do we understand by atypical lesions? Atypical lesions are such that the FISH probe, if it tried to detect digeorge, this particular deletion is beyond the scope of the probe to detect. So we might miss on those 10% atypical deletions. There are public data to support that SNP array was performed in FISH negative clinically suspected 22q11.2 microdeletion syndrome. Another question arises, is microarray an answer for all the structurally abnormal fetus? So Wagner in New England Journal of Medicine actually pointed out very important points. He made a mention that additional information over and above karyotype was provided by microarray in approximately 6% of fetuses who had anomalies, and these were clinically significant copy number variations. So when it was an isolated anomaly, the diagnostic yield of CMA was 5.6% versus when it was multiple anomalies, the diagnostic yield of CMA was 9.1%. Now, there's another paper by D. Witt in Ultrasound Obstetric Gynecology in the year 2014. He do mention that approximately 3.1 to 7.9% of fetuses with a structural ultrasound anomaly restricted to one anatomical site and a normal karyotype will show a submicroscopic CMA. Now, another question arises, which means which is the permutation and combination of organ anomalies which actually gives you a better diagnostic yield if subjected for CMA. This is the order, and this was actually described by Levy in Fertility Sterility 2018. So cardiac anomaly is at the top, followed by renal. Let us come to case 2. So we had a 24-year-old primary gravida at 16 weeks of gestation with a high-risk screen, and her nuchal translucency was within normal limits. So when we subjected, because the indication was high screen, she chose CMA over karyotype, and CMA was performed, and there was a deletion. If you can see over here, there was a tiny deletion. This is the normal copy number variation. You can see a dip over here, and this is marked in red. You can see a dip over here. That was a copy number variation, and it was deletion. So we diagnosed this as Mowat-Wilson syndrome after doing a literature search and figuring out that particular chromosome, that is on chromosome 2 on this particular region. This was the gene involved, and this gene, when lost, is primarily responsible for Mowat-Wilson syndrome. Then this lady who came up with an ultrasound which was normal is because most of the times in Mowat-Wilson syndrome, they can present with a normal prenatal ultrasound findings. However, in a proportion of cases, they do present with microcephaly, Hirschsprung's disease, cardiac defect, and distinct facial features. Kikoni also estimated that there was a recurrence risk for Mowat-Wilson syndrome, which was as high as 2.3%. Again, I re-emphasize that here two-parental study was required. Now the question arises, is this indication restricted to a structural anomaly, or how do we go about when it comes to CMA testing? Wagner in the year 2012 made a mention that approximately 1.7% of fetuses were detected to have microdeletions when the indications were other than the structural abnormality. He is very clearly mentioned in his, the other indications were the standard indications. They are advanced maternal age and high-risk screen. Another important point he has actually made note of is prenatal manifestations of microdeletions are yet to be established. In this particular paper, they have also mentioned that there was a baseline risk of 0.52% risk of detection of a submicroscopic deletion in women with an uneventful prenatal examination, which means that the screen was normal and there was no advanced maternal age. Then why was even an invasive testing done is the question. Invasive testing is done because of parental anxiety. In fact, about 900 plus patients actually underwent invasive testing due to parental anxiety, of which 0.52% were detected to have a chromosomal imbalance. Let's move on to case 3, elderly primary gravida with 16 weeks of gestation and a high-risk screen. So we detected a copy number variation in the form of gain, which is represented in blue. Okay. Yes, we are in a soup, because when we looked at the literature, we figured out that yes, this is a copy number variation. There was a difference, but this fell in the category of variant of uncertain significance. How do we go about? Now let us answer these questions. What are the risks of using CNA? What are variants of uncertain significance? How should they be managed? It is, of course, a challenging task to manage a variant of uncertain significance. So I have done a little bit of literature search and brought it up, but it is really challenging in practice. Copy number variations that are not commonly seen in the population fall under the category of either pathogenic or variant of uncertain significance. Coming to variant of uncertain significance per se, there are two important things which I've highlighted. One, there is little or no evidence to ascertain their pathogenicity. Second, they do not have a clearly established or specific clinical outcome. Now, points to ponder when we deal with variant of uncertain significance would be, let us look how small the duplications are. If these are small duplication, they're less likely to be clinically significant than the smaller deletions. Two, look at the genes which are present in that interval. Determine the parental status. This is of prime importance. So it has been said that if the CNV is de novo, then they are more likely to sway towards a pathogenicity. And if it is inherited from one of the parent, it will more sway towards a benign variant. But it is again not established. This is just an understanding. So what do? We go about counseling and we update with the current genome database to see, have there been more cases? Is there been a more of a genotype-phenotype correlation to establish whether variant of significance either falls in the pathogenic category or it falls under the benign category. This is my last case. Are all microarray platforms the same? We keep saying chromosomal microarray. So shall I subject the prenatal testing for a array CGH and not SNP? Or SNP and not array CGH? Why should I choose? What are the merits and demerits of these two? So we had a primate at 18 weeks of gestation who was found to have macrocephaly, intrauterine growth retardation, and oligohydramnios. And the microarray looked something like this. So there were 3 lines in the allele graph, 1, 2, 3, and 4. Normally we have 3 alleles, 1, 2, and 3. What are alleles? We have, let us look at it. Let us presume that this is chromosome 1 and it is homologue. Its homologue is a chromosome 1. Okay, so autosomes occur in pairs. So you have one gene located on one chromosome and you have its allele located on the second, on the chromosome 1, its homologue. So it can be A, or it can be A, B, or it can be B, B. So which means there are 3 lines. I'll explain to you in the text form. In a diploid, that is a normal individuals like us, we have our allele pattern which are represented as AA, BB, and AB. That is 3 lines. In a triploidy, allele status is represented in the form of 4 lines: AAA, BBB, AAB, and ABB. Now, the big question is, array CGH does not detect allele pattern, whereas SNP identifies allele pattern, which means if I had subjected that particular case for array CGH, probably I would have missed out on this triploidy and would have reported this as a normal array, but SNP identified allele pattern. So in the NEGM, so the author has mentioned that we therefore suggest that arrays used for prenatal testing should contain SNP probes that can reliably identify triploidy. Now, another question. Do we proceed with a karyotype in a prenatal setting when the array results are abnormal? So we do adopt karyotype of fish to detect parental balanced rearrangement in scenarios where the fetus was found to have an abnormality, since they have ramifications on the reproductive risk. Additionally, there's an important point I need to make a note of before I close my session. In case of trisomies of an acrocentric chromosome, that is chromosome 13, 14, 15, 21, and 22, which is detected by CNA, a karyotype should be performed. You know why? In order to rule out inheritance. Let me again re-emphasize, when it comes to a Down syndrome or when it comes to a Patau syndrome or trisomy 15, karyotype will easily identify this. Chromosomal also will easily identify this, but karyotype will tell you whether it is a structural translocation which has led to a trisomy or it is a free trisomy. Whereas microarray will only tell you there is a gain of 13 or a 14 or a 15 and accordingly. It will not tell you the mechanism or the arrangement the way the chromosome is. Why is this important? This is important because if I have a fetus who has a Robertsonian translocation which has led to a trisomy, I need to figure out whether this Robertsonian was inherited from one of the parent. So this again has ramifications on the reproductive risk and her future pregnancies. So with this note, I have kind of finished my cases and Dr. Mary will be actually dealing with subsequent cases, and she has many more interesting cases where karyotype has also been used. So I will hand over this to Dr. Mary.
Thank you, Dr. Vandana. So I'm Dr. Mary. I'll be continuing from here. Going on to case number 5. This was a 34-year-old lady at 17 weeks of gestation, and ultrasound showed increased nuchal thickness. For this indication, an amniocentesis was performed. Karyotype was normal, and therefore a chromosomal microarray was ordered. The array showed an abnormality. Specifically, there was a duplication on chromosome 7 on the long arm. And it was found that you can see the duplication here. You can see the duplication here in the peak on the graph representing chromosome 7. And it was found that this duplication was sitting in a region that was well known for copy number variation, specifically microdeletions that were causative for a well-recognized syndrome known as the Williams-Buren syndrome. So the Williams-Buren syndrome is caused by microdeletions in this region, and our patient had a duplication in the very same region. The question is it pathogenic? And there were 25 OMIM genes in this region. And we also found that there was a described syndrome that was associated with duplication in the segment. The syndrome was called the WBS duplication syndrome or the Somerville-Van der Rohe syndrome, manifestations of which included neurological and behavioral concerns with prominent speech delay, craniofacial dysmorphism, and aortic dilatation. The syndrome is actually, these manifestations are actually the reverse of what we usually see in the microdeletion syndrome, which usually manifests with microcephaly and aortic stenosis. In the duplication, we see macrocephaly and aortic dilatation. Most cases of this syndrome are de novo, but it can be inherited in a quarter of patients. And for this reason, we ask that the parents also be tested by chromosomal microarray because manifestations in the parents can be quite subtle and may not be recognized. But if the parents show the duplication, there is a possibility for recurrence. So that is why we ask that the parents be tested, and they were tested but were found to be normal. Prenatal identification of the syndrome is quite rare. So what are the lessons that were brought home by this case? One is about the pitfalls of targeted testing. Whenever we encounter an abnormality that is strongly associated with a particular chromosomal abnormality, for example, aneuploidy or a particular specific microdeletion or microduplication, we are very much tempted, especially in a scenario where economics is important, to go for targeted testing. But we have to remember that targeted testing is exactly that. It tests only for those specific targeted chromosomal abnormalities, and it can miss relevant genomic abnormalities that are sitting elsewhere in the genome. The other thing that we need to remember are that the prenatal manifestations of microdeletion and microduplication syndromes are largely unknown. In this specific case, I am uncertain whether the thing that we encountered was actually a sequence of the microduplication, or whether it was it was just a coincidental finding. But I'm sure that as we do further genetic testing, as the data accumulates, we will be able to define better the manifestations of the various microdeletion and microduplication syndromes. So if we're going to opt for a genomic testing, a test that covers the entire genome, we have the options of chromosomal microarray versus karyotype. And I think this study, Wapner's study, was mentioned by Dr. Vandana. This was a landmark study that was published in the New England Journal of Medicine in 2012. And as she mentioned, the study found that CMA identifies a greater percentage of pregnancies with clinically significant chromosomal abnormality, both in patients with structural abnormality and in patients with other standard indications for testing, such as increased maternal age or positive biochemical screen. And it was as a result of this study that the ACOG and the Society for Maternal Fetal Medicine, the SMFM, in 2016 made recommendations where they recommended chromosomal microarray analysis, or CMA, for prenatal diagnosis in cases with one or more fetal structural abnormalities. However, they also mentioned that in the absence of fetal structural abnormality, either CMA or karyotype could be offered. And this raises further issues because prenatal pickup will depend on the presence of features that can be easily observed by ultrasound. And like I mentioned earlier, many of these microdeletion and microduplication features, microdeletion and microduplication conditions do not have actually features that are clearly associated with them. And dysmorphic features that are a clear indication for testing postnatally are not always easily identifiable on prenatal ultrasound. Moreover, the features that we are looking for may not present until later in the pregnancy. So we are maybe missing a window of opportunity to detect something of relevance early in the pregnancy if we opt for a test with like karyotype that has a lower pickup. So this study was published in 2018, and it examines the recommendations of the joint recommendations of the ACOG and SMFM. And they examined over 3,000 patients and categorized them according to these recommendations, or as one category with structural abnormalities and another category that underwent invasive testing for some other indication. And they found that 2.5% of patients with a structurally normal fetus who underwent invasive testing would have a clinically significant structural chromosomal abnormality that could be missed on karyotype. That means 1 in 40 patients who have who undergo invasive testing for an indication other than structural abnormality in the fetus are likely to have a significant chromosomal abnormality missed if they were to opt for karyotype instead of chromosomal microarray. And that's something to think about. So I'm going on to my next case. This was of a 43-year-old patient at 18 weeks of gestation. The fetus was detected to have clubfoot on ultrasound, and amniotic fluid was subjected directly for chromosomal microarray analysis. We found abnormalities on 2 chromosomes. On chromosome 16, there was a small terminal deletion on the short arm. On chromosome 17, there was a much larger here it is, a much larger duplication on the long arm of chromosome 17. So to mention once more or to reiterate, there is a deletion on the short arm of chromosome 16, and this deletion included 35 OMIM genes. And at the same time, there was a gain on chromosome 17, and this duplication contained 152 OMIM genes. This is how it is shown on the chromosomes. It is represented on the chromosomes. This is the deletion on chromosome 16. It's terminal. That means it's at the end of the chromosome. This is the duplication on chromosome 17. Again, it's terminal. It's at the end of the chromosome. Question is it pathogenic? And we found that there were described syndromes related to each of these abnormalities. There was an alpha thalassemia mental retardation syndrome, chromosome 16 related, or ATR16 syndrome, that mapped to the region that was deleted on chromosome 16. And at the same time, there was a distal trisomy 17q duplication syndrome that mapped to the segment that was duplicated on chromosome 17. So definitely it was pathogenic. Another feature of the chromosomal microarray that I would like to highlight here is its ability to clearly delineate or define the genes that are involved, because there are other syndromes that also map to the same chromosomal band and to the same chromosomal band, and there are other genes that map to the same chromosomal band, and because the chromosomal microarray defined the genes involved, we were able to say that these particular genes were not in the involved segment. So we were able to clearly define what was the syndrome, that potential syndrome that the fetus would be suffering from. And that is important for genetic counseling. Now here we had a deletion and a duplication involving terminal ends of the two chromosomes. And whenever you get this situation, you have to consider the possibility of an unbalanced translocation, possibly arising from inheritance from a parent who is a balanced carrier of such a translocation. So to elaborate further, here we have a parent who is carrying a balanced translocation. The chromosome 16 segment of the chromosome 16, the short arm, and the chromosome 17 long arm have interchanged positions. The parent is not affected because there is no gain or loss in the parent's genome. So the parent is unaffected, he's perfectly normal and healthy, and is quite likely unaware that he carries such an abnormality. But during meiosis, when the chromosomes segregate, this can be transferred to a gamete in an unbalanced fashion, and to the zygote that results, or to the offspring that results from that gamete. This will result in chromosomal imbalance. So here we have the child who has inherited the abnormal chromosome 16 and the normal chromosome 17. So what has happened is that the child has a gain of chromosome 17, of a segment of chromosome 17, and a loss of the segment of chromosome 16. So this is what has happened in this particular case. And it's important to think of this possibility and to test the parents. Like has been mentioned before, you cannot test the parents using a chromosomal microarray, because chromosomal microarray will not detect balanced translocations, balanced abnormalities. So you have to test using karyotype. And it's important to do this because the parent still carries the abnormality and there is a risk of recurrence in future pregnancies. And this recurrence could manifest not only as a fetal abnormality, it could manifest in the form of recurrent pregnancy loss, or even a live-born child with abnormality. So the lessons we learned from this case are that chromosomal microarray can detect and localize loss or gain in the genome, but it cannot define the mechanism. So sometimes you have to go back to karyotype or FISH to detect such a balanced translocation. And secondly, when there is loss as well as gain involving terminal ends of the chromosomes, you have to consider the possibility of balanced translocation in the parents, and you have to investigate for this, because otherwise we would be missing the chance of preventing a recurrence. So going on to my final case. This was a 31-year-old female, a fourth gravida with 2 living children at 13 weeks of gestation. Her screen risk was increased. That is, it was 1:60. And we did a karyotype of the chorionic villus sample and found a mosaic karyotype. Mosaic karyotype refers to a situation where there are 2 cell lines. So usually we look at 20 metaphases, and we found that in 13 of the patients of the metaphases from the sample, there were 47 chromosomes, including a supernumerary marker chromosome. A marker chromosome is a chromosome, the source of which cannot be identified. And in the remaining 7 metaphases, the karyotype was perfectly normal. So this was a mosaic karyotype with a supernumerary marker chromosome. And this is what it looked like. You can see the marker chromosome here. It's quite small. So there are no defining features, nothing that can actually tell us which chromosome this marker originated from. And the problem here is that we are unable now to decide on the clinical significance of this marker, because sometimes a marker can be harmless in a few situations, and sometimes it can carry severe clinical manifestations. So here we are not able to identify where exactly this marker has originated from. So we were in a bind. Most markers originate from a few selected chromosomes. So we used FISH as well as QF-PCR to interrogate and find out whether they originated from these chromosomes, that is 15, 22, 13, 18, 21, or 13, 15 or 16. And we found that both of these tests were normal, and parental karyotypes were also normal, because if the parental karyotype also showed the marker, we would have been reassured that most likely it is normal, because the parents also show it and they don't have any particular defects. But here the parental karyotypes were normal, so we were again stuck. So finally we decided to do a SNP array, and the array showed a gain of 41 megabases involving the pericentromeric region of chromosome 1. So the array told us that this was derived from chromosome 1, and it also showed us the region where it was derived from. And we wanted to confirm that by FISH, and luckily we had a whole chromosomal paint for chromosome 1. So we did a FISH using the whole chromosomal paint. And you can see here that these are the 2 normal chromosomes 1, and our little marker has also fluoresced. So it is also derived from chromosome 1. The question was, is it pathogenic? Now, because it was derived from the centromeric region, which carries heterochromatin, what is known as heterochromatin, which shows up as the dark region near the centromere on G-banded karyotypes, or is it the euchromatin, which is the light staining region on G-banded karyotypes? And it's important to differentiate, because heterochromatin consists of repetitive sequences with redundant genetic material, not usually functionally significant, and is usually not covered on an array. But if you remember, our marker showed up on the array. So that told us that it was not merely heterochromatin, and actually the delineation on the array showed that it extended beyond the heterochromatin region into the adjacent euchromatin. And we were also able to define the region, because the microarray defined for us the region that was involved. We found that it overlapped with a duplication syndrome known as the 1q21.1 duplication syndrome, manifestations of which included behavioral psychiatric manifestations like autism and schizophrenia, mental retardation, seizures in some patients, dysmorphism, and other congenital anomalies could also be seen. But the penetrance is incomplete. When I talk about penetrance, it refers to the probability that the patient will, that a patient carrying this abnormality will actually manifest with the clinical symptoms. So the penetrance being incomplete meant that there was a possibility that the patient or the baby could be asymptomatic. Anyway, the parents decided to continue with the pregnancy. The baby was born after LSCS at term, and the baby is now 6 months old and has achieved milestones appropriate for age, but it's still early yet. So we don't know what will happen in the future. So the lessons learned here were that even though we talk about the expense of chromosomal microarray, actually it can be a cost-cutting measure, because sometimes when you go directly to a microarray, when you find something that you can't really identify or define on a karyotype, instead of fishing around using other tests, you can save time and money by going directly to a chromosomal microarray. And the second thing is that chromosomal microarray not only helped us to identify the marker, but it helped us to delineate the region involved, to define the region involved. And this helps us to assess the clinical significance, which is so important for counseling. So those were the lessons learned, and that finishes my presentation. I'd just like to reiterate some points. One is that chromosomal microarray has a better yield for clinically significant chromosomal abnormalities than karyotype, and it should be used for diagnostic testing in pregnancies with structural abnormalities. And chromosomal microarray should be considered and discussed as an option in situations where invasive diagnostic testing is being done for other indications. And it also helps in accurately defining the regions involved in copy number variation, which is useful to determine the clinical impact. However, karyotype and FISH are not eliminated entirely. They still have a role in identifying, detecting balanced translocations and balanced chromosomal abnormalities. Thank you.
Thank you. Thank you, Dr. Vandana and Dr. Mary. Excellent presentations. You have actually brought out how chromosomal microarray is being used and what are the clinical applications, what are the implications of testing, what other tests can supplement your clinical findings, and also to give risk of inheritance. So I think we will now move on to a couple of questions which are in the chat box. So one of them is, how sensitive are NIPT offering microdeletions and duplication testing? Dr. Pradhan, do you want to take that?
NIPT in microdeletion and microduplication, it's not in routine use. And Dr. Priya, probably we will cover that in our last session.
Yes. Yes.
That will be NIPT and extended NIPT.
Yes.
That will be covered, the microdeletion and duplication will be covered in that session.
Yes.
So please do join us for that session.
Yeah. Then there is one other question. I think you've answered this, right? The recurrent rhombencephalosynapsis. Yeah. Yes. So you have recommended that they go in for an exome, clinical exome testing. Yes.
Yeah. So thank you, Dr. Vandana and Dr. Mary. That was nice series of clinical cases. And the main thing is that it's clear, crystal clear of explaining. From the cases, we learn quite a lot. And that also emphasizes the hybrid technology. I'm not talking about hybrid of Ayush with this thing, but hybrid of lab persons, with a cytogeneticist and obstetrician, and even ultrasonologist will tell you that there is a clubfoot, then you do it and then tell it. So a lot of hybrid now going on. So probably this is the best hybrid that we can have, all of us together on one platform. And thank you for doing this honor for this genetics and field medicine session. Dr. Priya, anything to
add on? Yeah, I think we just have, Joseph, if we can just have the last slide. Yeah.
Joseph will, before that, let me do this. And that next session will be on 6th of January. And there we will be trying to cover infertility and reproductive outcomes and genetics of it. Why I proposed this genetics of infertility was that a lot of things goes on. People go on doing the whole exome for infertility, then whatnot. So let's clarify it and follow a guideline. And probably at the end of it, if we as a committee and then FOGSI can formulate a guideline which tests to be done for a set of conditions. And knowing that how to interpret it is our goal of doing it. So, and subsequent to that, we will be doing on single gene disorder and including NIPT, and hope that you join us. I thank all of you, those who have joined. Probably 178 was participants. And I hope that more people join us, so that Dr. Vandana's last slide was excellent, awareness and acceptance. So awareness, you make us aware, and then let us accept this technology. Okay, thank you. Thank you, everyone.
Thank you so much. Thank you all participants. We will see you on 6th of January.
Okay, thank you.
So on behalf of MedGenome, I'd like to thank FOGSI and Dr. Mandakini Pradhan for providing us this opportunity to conduct this webinar. And I'd also like to thank all the chairpersons, Dr. Malhotra, Dr. Besar, and also the speakers, Dr. Mandakini herself, Dr. Shaktivel, Dr. Vandana, and Dr. Mary for taking time off from their schedules to give us this talks. And I'd also like to thank the audience for taking time off from their schedule, registering and attending this sessions. As I mentioned, our next session is on Jan 6th. We will be sending out invites for that webinar in a couple of days, and you can register using the same process and log in at that time. If you have any more questions or comments, you can contact us at the information, the contact details given below, and we'll be happy to address them, anything to do on chromosomal microarrays. And we'll be sending out a recording of this in a couple of days, and also be sharing it with FOGSI in due course. So once again, thank you for your time, and stay safe. I'll be ending the session now.
Thank you.
Thank you. Thank you, Joseph.
Everyone.